Okay, so I'm going to speak about Begomovirus disease. And as it was just introduced by my colleague, I will not spend much time on the virus, but I'm most interested on Betasatellites. So I'm working in Montpellier. So I'm working in the south of France, not far from Mediterranean Sea. And this is our building at Montpellier. And I'm working in a French unit, which is Plant Health Institute of Montpellier. And in this institute, I'm working in a team which name is Characterization and Modeling of Epidemics. And we are working on different we are studying different steps of epidemics, from the evolutionary of viruses until the sources of viruses, dynamics, until the evolutionary consequence of management strategies. So in this team, we have three main models. So the one is Cica virus on prunus, which is a quarantine virus in France. We are also working on viruses on cereal, which are very important, and the vectors now cannot be controlled because the pesticides are forbidden. So there is a real challenge to control these viruses. And then the one that I prefer, the tomato yellow leaf curl virus on tomato, which is present in all the Mediterranean basin. So, as you know now, begomoviruses are very important. They affect a lot of type of crops, and from cassava, cotton, and tomato and vegetable crops. And they were detected around the 80s, and they are transmitted by the whitefly Bemisia tabaci, which is present in all the intertropical area, and they are encapsulated in Gemini particles. And they can have monopartite or bipartite genomes. And so this is the geographical dispersion of these diseases. And at the end of the 90s, some scientists discovered that some viruses could be that they found were not, they could not reproduce the disease because they lack a component. And then it was mainly on cotton disease that they found that this disease could be reproduced, could be inoculated to plants only if the virus was associated with a molecule which is called a satellite. So a satellite is a mobile genetic element deficient for the genes of replication and encapsulation. So it's not a virus. It doesn't have any homogeny with the virus. So it's something which is not independent. It needs a helper virus to be replicated and be transmitted. So it's like a parasite of a virus. It parasites the function of the virus to be replicated in the plant, accumulate, and be transmitted. It's encapsulated in the same capsid protein of the virus, not with the virus, but in a independent capsid, and then it is transmitted with the virus by the same whiteflies. And its genome is composed of only one gene, which is beta-C1 gene, which is the determinant of function of these molecules. Begomoviruses have three different types of satellites, but the most important one is the beta satellite, which has this gene, because it increases the symptoms of the disease, the viral disease. And it is just this gene which is responsible of this increase of the symptoms. And also, it can increase the accumulation of the virus because it suppresses plant defense system and then allows a higher accumulation of the virus. So, the dispersion of the geographical distribution of these molecules are mainly in West Africa, Asia, until east part of Asia. And there is no satellites in the Mediterranean basin except in Egypt and now in Israel and Jordan. And there was arrival of beta satellites in the US with cotton, but then it was eradicated. So the beta satellites, there are two types of association with viruses. In some, as I told you, like a malvaceous plant, like cotton, the satellites must be there for the virus to infect the plants because the the virus cannot go over the defense system, so it needs the satellites to infect. So in this time, the satellite is like a viral component. It's not a viral component, but it acts as a viral component because the virus needs the satellite. But for the other viruses, Begomovirus, there is no need of this satellite. But they can replicate the satellite. So they replicate the satellite even if they don't need it. And sometimes it increases the symptoms of the virus. And then it creates a new disease, by fact. And so you have this, okay, so there is two types. So you have these mandatory associations which maintain the satellites because the satellite has to be transmitted for the virus to infect. So in cotton, for example, you always have the satellites. If the virus is there, the satellite is there. So this system maintains the satellites. But in tomato, for example, with tomato leaf curl New Delhi virus, for example, you don't need the satellite. The virus can infect by itself. So you have these two systems, and this raises some question about how can this satellite be maintained when the virus doesn't need it. And if you see in the literature, there is no specificity between the virus and the satellite. So we have now around 400 Begomovirus different and 100 beta satellites, and maybe all of them can be replicated by all. So this makes a very huge amount of association, which can be dangerous. So this is a very big problem for Mediterranean area where you have a lot of vegetable crops. We have 6 different Begomoviruses affecting mainly tomato and cucurbits, and we are very afraid of the introduction of satellites there. And in 2012, yes, thank you. In 2012, a beta satellite was detected in whiteflies in Jordan, but the disease emerged only in 2016. And we don't know what happens between this time. And when the disease emerged, the scientists there, they sequence the beta satellite and say, oh, we have a beta satellite. They look in the literature and found that this beta satellite comes from Israel. And they said, but we didn't have it. But it was published by someone else who took some samples and found the beta satellite in 2012. But the disease appeared only in 2016. So now we are really afraid because so it's there, it's in West Africa, and we think that it will arrive in the Mediterranean basin because of movement of whiteflies, intensification of vegetable crops. So what we are studying at Montpellier now is we have begun some studies to prevent this emergence of new disease, and we are studying the capacity of maintenance of beta satellites by a virus which doesn't need the satellites, because the virus we have here, these viruses, they don't need beta satellites. So we have studied the capacity of maintenance of beta satellites by these viruses, the dynamic of accumulation in planta, the cost for the virus, and the transfer of beta satellite between hosts and vectors. So this is the first thing that we have done to see how they can be maintained by our viruses in the Mediterranean basin. So we use agroinoculation methods of clones, and we inoculate beta satellites isolated from okra in West Africa, which is cotton leaf curl Jezira. It's an error, beta satellites, not a V, but a beta satellite. And then we inoculated tomato plants and follow the infection rates and accumulation, DNA accumulation. And we found that with the virus, we know that we have these type of symptoms with our TYLCV, tomato yellow leaf curl, and tomato yellow leaf curl Sardinia virus. But when you add the satellites, then you have very important symptoms. You can see here the size of the leaves between the healthy plant, the virus, and the virus plus the satellite. So we have 100% transmission of the satellite with the virus, 90 and 50. So it depends on the virus. And then we did it also with New Delhi virus, because New Delhi virus was introduced in Europe in 2012. And it's the strain which was introduced in Europe could infect only cucurbits, not very well tomato. And so we tested how this satellite could work with tomato leaf curl New Delhi, and then we did the same experiment. And then here you have the disease of you have the healthy plants, the plants with the virus, melon and zucchini and melon. And when you add the satellite, you have very severe symptoms. And then you have also here the size of the leaf. So this beta satellite increased our symptoms, viral symptoms. Then we did the same thing with another satellite that we found in Iran, which was isolated from a papaya plant co-infected with TYLCV. So we did the same thing with our virus, and then we saw that this beta satellite could be replicated by the tomato virus, but not very well with TYLCV Israel strain, but not with these two viruses. And it could not be replicated by tomato leaf curl New Delhi. So you see, it depends on the virus, it depends on the host, and it depends on the satellite. So you never know what will happen. Then we follow the dynamic of accumulation of satellite with our virus, which is tomato yellow leaf curl virus. And then we found that this is the dynamic of the virus alone in the plant. And when you add the satellite, so you see that the virus accumulates at a lower level when the satellite is there. So there is a cost for the virus. And the satellite began at 11 days post-inoculation. And from 18 days post-inoculation, its accumulation is higher than the virus. So there is a very high amount of the satellite in the plant. Then we checked until 150 days post-inoculation. And then you see that at 150 days, post-inoculation, the amount of satellite decreased. So we thought that maybe at the end of the satellite will disappear, because after that it will not be transmitted anymore. So we tried to see if it was transmissible. So we took some plants which were 32 days or 150 days post-inoculation, and this is the virus accumulation alone or with the satellite. At these 2 different days, and we did transmission test, and we found that the satellite could be transmitted very well, even at the very late stage of infection. So this means that it's not because it decreased in accumulation that it's not transmitted anymore. Then we thought that maybe we know that in Europe TYLCV doesn't infect zucchini. But since this beta satellite can overcome the resistance, the gene silencing in the plant, we thought that maybe it could infect zucchini when it is associated with the satellites. So we tried co-inoculation of both, and we did inoculation by mechanical agro-inoculation or vector inoculation. And then we thought that it wasn't possible. So the beta satellite doesn't help the virus to extend its host range in this case. Then we know that tomato leaf curl New Delhi doesn't infect tomato efficiently in our country. So we said, okay, maybe with the satellite it will be better. We tried the inoculation with the beta satellite, and we observed that it was not better. So this beta satellite doesn't enhance the infectivity of New Delhi virus in our country. Then the last thing we did was to see if the satellite could be transmitted from one virus to another virus in a different host. Imagine that a beta satellite arrives from cotton. So it is collected by whiteflies in okra plants, and we know that this virus doesn't infect tomato. So if this virus, Bemisia, arrives on a plant which is already infected with TYLCV, can it become TYLCV plus beta satellites? That means that the beta satellite could be transferred. So we tried and we obtained, in 50% of the cases, the experiment was done twice, and in 50% of the cases we obtained plants which have the beta satellites. Then we say, okay, we have insects which have collected this virus from okra, the virus from tomato, and the beta satellite. If we put it on okra plant, can it transmit? It's the right couples, means Jezera virus and beta satellite. We tried, and yes, we obtained 100% of infection of okra plants. And on tomato, we did the same thing, and we observed that 75% of plants could have the virus and the beta satellite, the TYLCV and the beta satellite. So we were able to dispatch the different couple in the good plant, in the right plant. So this means that the beta satellite can be transferred by whiteflies with the good helper virus. Then, in conclusion, so we have seen that Begomovirus of the Mediterranean basin can maintain beta satellites, depends on the host and the satellites. And this has some impacts on virus accumulation and symptoms, depending on the association. And that whitefly transmission is efficient even at the late stage of infection. This means that we have to be very to make a survey, regular epidemiosurveillance in this area, as well in whiteflies and reservoirs, to be sure that we will not have introduction of these beta satellites, because sometimes we may not see them, because they can be there without being seen, and as it happens in Jordan and Israel. And also, what we are afraid is the capacity of beta satellite to adapt to new viruses, because I told you that maybe sometimes beta satellites were not replicated by the virus, but in some case it could be progressive, and then maybe after a few crops we will have adaptation and replication by a virus which was not able at the beginning, maybe after mutations and co-evolution of the satellite and the virus. And this has already been shown once, and we have to be in another country, and we have to be very cautious to this. And then beta satellite can be transferred between Begomovirus and host plants, even if the virus, which is the helper, the first helper, cannot infect the new host. This means that the satellite it is as if for us the satellite can move alone, because we don't care the virus he comes with. So we are now working on this to see if a virus can even, Bemisia can even inoculate a plant with the satellite alone, and the satellite will wait that a virus arrives. Because we have seen that if the virus is there, it can replicate the satellite. But if the satellite arrives with no infecting virus, can it wait like a component and wait that the virus arrives? So this is what we are working on now. And then what we are also doing is to try to modelize the propagation of a beta satellite in a new environment by using this system, which is a mandatory system with a virus which needs the satellite, and the rest of the environment which doesn't need the satellite, and to see how it can work and if the satellite will be maintained or not. And for this we will need a better knowledge on beta satellite epidemics in infested area, and we know that in India you have a lot of beta satellites. There is very few epidemic studies because many people are working on molecular biology, functioning of the beta satellite, how they interact with the plant and the virus, but there is almost no epidemiological studies about telling us, for example, this satellite is with this virus and not with this one, and it moves from this plant to this one. We don't have these ideas and this information. This is what we are interested in. Maybe if there are some, it could help to develop, to modelize and have a better knowledge of the disease, of the risk that we may have in the next few years. Okay, that's the end. So my research to the team I'm working with, and this is Montpellier and the seaside. And this is the project that we have worked with, and the Region Occitanie, which paid for a lot of help for students with transmission, vectors transmission, and a private company which produce resistant seeds.
Long live India and France friendship.